Hi, so I'm Shu, a clinical fellow. Today, I will talk about novel immunotherapy in cancer therapy. In cancer, some of you may wonder why I'm choosing this topic, but actually, this is one of the hardest topic in any cancer field right now. So you will hear a lot about this in a few years' time in urology as well, and so. I want to guide you through this very interesting topic, even though I have no background in immunology, just to let you know. Okay. So today I'll talk about first basic mechanism of immune tolerance, how it works, and there is a pathway called PD-1 PD-L1 axis, which is actually the target in this new therapy. So I'll talk about that, and I will then go over pre-existing phase 3 very strong data in other types of cancer, and in the end I will talk about what's happening in the urologic cancers as well. So here are some fancy titles from recent papers. When I go through all of the top journals, Nature, JAMA, New England, everything has published reviews on this immune checkpoint blockade in the last several years. So it's really the hottest topic in cancer. So first of all, I want to go briefly through this immune tolerance. And usually for T cells to be activated, first there should be antigen presentation, specific antigen presentation by antigen presenting cells or sometimes by cancer cells. And this is the main signal, which is antigen specific. However, T cells cannot be activated enough to do something just by this main signal. There are co-signals that are actually important for regulating the fate of T cells. And this seems to be very context dependent. So main signal is antigen specific, but co-signals are rather antigen non-specific, and it's context dependent. And this one actually determines the fate of T cells. So the famous ones are here CD28, which is important for activation of T cells. And there are ones called suppressive signals. The most well-known of those are CTLA4 and PD-1, that both of those which I'm going to talk about today. So immune tolerance is induced in the presence of negative co-signals. So if even if there's antigen-specific signal presented, if the co-stimulate is negative, then T cells won't be activated and immune tolerance is induced. More specifically in cancers, in the lymph node, when so cancer's peptide from cancer is presented by antigen presenting cells. In lymph nodes, mostly it's the CTLA4 that's the suppressive signals. So there's CD28, which is activating signal, and CTLA4, which is a suppressive signal. And if this overcomes the signal of CD28, then T cells won't be activated. PD-1 acts by PD-1 PD-L1 acts more peripherally. So in addition to some function in the lymph node, it's more in the tumor microenvironment where PD-1 and PD-L1 interaction occurs. So in addition to antigen presentation by APCs, tumor itself express PD-L1, and if this signal is T cells catches this signal, then this T cell is not activated. So T cells won't attack the tumor cells if tumors are expressing this PD-L1. Just to, before going any further, I just want to show the history of the development of this PD-1, PD-L1 targeting therapy. PD-1 was first identified in 1991 by Dr. Honjo, actually from Kyoto, where I come from. He used to be the dean of my medical school when I was a student. I didn't know at all, but first, they were studying about apoptosis mostly. And PD-1 is actually programmed death 1. So they thought it was this molecule was associated with apoptosis. Later, it was proven that it doesn't really have much function in apoptosis, but that's how it was first identified. And it took about 7 years for them to find out that its function is more in the immune system rather than the apoptosis. They they created a mouse model lacking PD-1 and closely watched the phenotype, and they actually find out that mice lacking PD-1 develop autoimmune disease. So and later they found that it's very important in immune tolerance. And around that time, they went on to identify the ligand for PD-L1. So it took about 8 years or so to find its role and function, its molecular function. So PD-L1 binds with PD-1 and induces immune tolerance. And when they found about PD-L1, they went on to stain many tissues and found out that indeed cancer cells itself, lots of cancer cells express PD-L1, and that the expression of PD-L1 is associated with the outcome of cancer. So I will show you later, but cancers expressing high level of PD-L1 tend to do worse because they attain immune tolerance, and they won't be attacked by T cells, resulting in worse prognosis. And so they got the patent, and they went on to develop a monoclonal antibody that can be used in the clinics. And in 2010, first phase 3 trial using CTLA-4 antibody was approved for melanoma. There are 2 phase 3 trials came that showed survival benefit in melanoma. And in 2012, phase 1 trial was completed for solid tumors, and this included renal cell cancer, which showed a very good response. And in 2014, PD-1 inhibitor was approved cleared the phase 3 trial and approved for melanoma. And just last year, I will talk in detail about this study, but a PD-L1 inhibitor actually showed a significant activity in metastatic TCC, heavily treated chemotherapy-resistant TCC, and it's got FDA breakthrough status granted, and now is just recently started the phase 3 trial in bladder cancer. And just a week ago, when I was going through all this phase 3 trial, it's not published yet, but phase 3 trial for non-small cell lung cancer came back positive. They beat this PD-1 inhibitor, outperformed docetaxel in metastatic non-small cell lung cancer, and it was just recently FDA approved. So we know that it works just not in melanoma, but in heavily treated solid cancers as well. So going back to the original discovery, so they discovered PD-1 and then created PD-1 knockout mouse. And this is what it shows. So when we knock down PD-1, this is the glomerulonephritis, many infiltrating cells, and also this is arthritis where many immune cells invade the joints. And also PD-1 knockout mouse developed autoimmune dilated cardiomyopathy. So this is the wild type. This is the PD-1 knockout mouse, which shows significant dilated cardiomyopathy. Just to let you know, so this PD-1, as I've told you before, acts more peripherally. So their function on the system is more dependent on the context. It's not really lethal by itself. But when we knock down CTLA4, which acts more centrally, the effect is larger. So CTLA4 knockout mice usually die within 5 weeks due to severe autoimmune disease. So these phenotypes in mouse show difference in central immune system and peripheral immune system. And not only the autoimmune aspect, but PD-1 also affects the antiviral immunity as well. So when mice are transfected with adenovirus, these blue shows virus. So in wild type virus infected, the liver is infected with virus, but in PD-1 knockout mice, since there's no suppressive break from PD-1, there's immune system is largely activated, balanced on the active side, so the virus is cleared very rapidly. So these basic data show that PD-1 pathway is also important in immunity as well as immune tolerance. So after that, they went on, as I mentioned, they went on to find PD-L1, and people started to look at PD-L1 expression in cancers. This is the data for renal cells on 306 samples. PD-L1 high has worse survival. This is urothelial cancer, the same data. And this is ovarian cancer, looks the same. Similar data in other cancer cell types as well. So overall, in many cancer cells, cancers expressing high level of PD-L1 has poor outcome. And I think the beauty of this PD-1, PD-L1 axis immune tolerance story is that it was based upon the from basic research, and it was step by step for it, and now it's in the clinics. But when I look closely into its biology, especially in the setting of cancer, there's not really much data there. I think the reason is because to study those, you need an intact immune system. So you cannot use cell lines, you cannot use xenoimmunodeficient mice. You have to think of the way, the model is very limited. But still, there's some data, and this is one of them. So in melanoma, there are cells that can metastasize even in not only in immunocompromised mice, but regular mice. And when they instill those cells in wild-type mouse, they metastasize, but in mice with PD-1 knocked out mice, there's less metastasis. And also the same thing, same phenotype was observed when they use anti-PD-1 antibody as well. So either way, grown in PD-1 knockout background or using anti-PD-1 antibody, suppression of PD-1 results in less metastasis in melanoma cells. So on these, from these data, now they've kind of started to work on phase 1 trials in humans. Next I will talk about human data, but before going on to human clinical trial data, there are certain things you should know before reading those papers. One is this immune-related adverse events. Unlike chemotherapeutics, these immune checkpoint inhibitors have specific side effects called immune-related adverse events. Usually, this occurs by non-specific immunologic activation of the immune system. So the phenotype is similar to autoimmune diseases. So common ones are dermatologic, like skin rash and itchiness, or diarrhea, elevated hepatic enzymes, and some rare but some endocrinopathies. And also some severe pneumonitis is reported, although the incidence is very low. And of course, it's activation of immune system that's causing these side effects. So the treatment is usually steroids or immunosuppressive medications. So this is totally different from the what we usually see with chemotherapy. The other thing we should know is that this pattern of response is a bit different from those seen with chemotherapeutics. So this is straightforward, going down just straight down. But these are actually the patterns of response shown in melanoma phase 3 trials. Some tumors actually increase a bit, although so have some period of stable disease and then decline over the time to become PR. And there are some cases, more extreme cases, where they progress in the first 3 months and then show response, almost PR to partial response to complete response. And also, this dotted line is new lesion. And originally, usually in RECIST criteria, when we see a new lesion, that's automatically called as progression. But in these immune response criteria, even if a new lesion appears, they seem to also respond to therapy. So new lesion does not necessarily mean progression. So now they came up with immune-related response criteria, which was actually variated to correlate better with survival in the phase 3 melanoma trials. The biggest difference is that, as I mentioned, in this new criteria, new lesions is just added to the tumor burden. It's not, it doesn't mean progression. It doesn't necessarily mean progression. It's just added to the tumor burden of the measurable disease. And also any progression requires confirmation. So originally with RECIST, any progression at just one point was enough to call progression, but in this criteria, any progression requires confirmation because there are some odd response after initial progression. So I'll move forward to activity and safety of PD-1, PD-L1 inhibitors in human patients. This is the initial data I talked to you about. It first came in 2010 with CTLA4 monoclonal antibodies. And two phase 3 trials are both showing improvement in overall survival. As in other medications, usually it's like 3 months or so. Recently, this second paper was, follow-up of this second paper has been published, and even though there was only 2.1 month overall survival difference here, when we look at 5-year survival rate, there's double difference in overall survival rate after 5 years. So of the 250 patients in each arm, patients receiving this CTLA4 inhibitor had 40 patients alive, whereas with placebo it was only 20. So this is quite significant, considering that melanoma is a very, very, very bad disease in the metastatic setting. And this is the most recent report which came last year. This is a PD-1 monoclonal antibody showing similarly survival benefit in melanoma. And this is quite striking. This is the overall survival. So, and they compared directly against the chemotherapeutics dacarbazine against both single agents against chemotherapy. And with conventional therapy, median overall survival is 10.8 months. And with this PD-1 inhibitor, it's not even reached. It's a quite huge difference here. And this is progression-free survival. This is the waterfall plot. You can just visually see how much difference there is. And also, we know from the other studies as well that one of the good things about these inhibitors is that its duration is quite long. So unlike we know with many anti-angiogenic inhibitors like sunitinib or sorafenib, that their duration of response is not really very long, but with these, it's really long, and here duration of response is not reached for this anti-PD-1 as well. Looking at the side effect, when we compare to this chemotherapy, actually the incidence of adverse event is indeed lower, and it's the specific ones like pruritus and rash is a bit higher for this PD-1 inhibitor. As I mentioned, there's specific side effects. But if we compare this nausea or neutropenia, thrombocytopenia, there's no hematologic side effect. So it's much more tolerable than these conventional chemotherapies. And now moving on to the solid cancers. In 2012, both PD-1 data and PD-L1 inhibitor data came up at the same time for solid cancer. And luckily, they both included renal cell cancer, presumably because renal cell cancers in some aspects act similarly to melanoma. So, and this is the objective response rate. They included 66 renal cell cancer, and objective response rate was 27%. which is quite high, considering that most of these patients had very high burden metastasis, and they had most had been previously treated with some kind of systemic therapies. And this is one of the examples which shows a very huge, I think this is lymph node rather than primary, but this huge mass with renal cell cancer the size decreases to this much after 6 months. And for PD-L1 inhibitor, it also has some effect. Although in this initial trial, the effectiveness was, objective response rate was a bit lower for renal cell. It was 12%, but still in many cancer types, it had around 20% response rate. And in some cases, it's very durable, long-term. It acts for long-term. And here, this is non-small cell lung cancer with liver metastasis, which is usually it was thought to be untreatable, but with this therapy, it almost diminishes. So they have shown clinical activity in solid cancers as well. And what happens if we do combine? So I told you that PD-1 acts at periphery, and CTLA-4 acts at the central immune system. So they tried combination of total blockade of checkpoint inhibitors, immune checkpoints, and 53% of patients had tumor reduction 80% or more in melanoma. This is quite striking response. Although grade 3 or 4 adverse events occurred in 53%. So it's a bit more toxic. They say they were generally reversible, but it's a bit high, I think. But addition has its own benefit. And now there is a phase 1-2 trial combination trial going on where bladder cancer is one of the cancer types that's being tested.
So in all these clinical trials, is the drug always on board? Or are there clinical trials actually that show what happens to the tumor when the drug starts?
Yeah. There's people talk about memories as well.
These clinical trials are with the drug on board. They never stop it.
For these, I think there are cases, follow-up studies that they did try to see, and there are a couple of reports I read that even after stopping these medications, yes, effect goes on for some cases with immune memories. Yeah. And this is the breaking news, just the one I mentioned. FDA approved this PD-1 inhibitor for squamous non-small cell lung cancer. So the phase 3 trial showed against docetaxel showed 41% reduction in the risk of death. And this was interim analysis, but already there was median overall survival of 9.2 months versus 6 months. So now they're allowing patients to switch over to the anti-PD-1 given this data. So it's quite significant. So now let's move on to the current status in urologic cancers. First, renal cancer. Just recently, a phase 2 trial for PD-1 inhibitor was published. They tested in 168 patients, renal cell cancer patients, with 3 different doses, and it's given every 3 weeks, 60 minutes IV. And primary endpoint was dose response assessed by progression-free survival, and they had these secondary endpoints. And looking at patient characteristics, about 25% are poor, fall into poor risk in MS cases, memory sloan Kettering risk criteria. And many of them have high burden disease like liver mets. And about 70% of patients were treated with 2 or more systemic therapy, including anti-angiogenic or mTOR inhibitors. So there are really many bad patients included in this trial. This is the response rate. The response rate was overall about 20% in all the doses. And there was actually no significant difference in response rate comparing these 3 groups. However, when we look at overall survival, there was, although still not statistically significant, a higher dose tended to do a bit better with overall survival. And this is the duration of response. This is month. So many patients, this is about 2 years. So many patients have a durable response more than 2 years. And median duration of response has not been reached in 2 doses. And at data cutoff, 40% of the patients who had partial or complete response were still responding to therapy. So response rate is about 20%. Not all patients respond, but if for those who respond, it's really durable. And the side effects, they studied immune-related side effects. This is treatment-related, so immune-related side effects and other side effects as well. And overall, it was all 3 arms were tolerable, even though systemic corticosteroids were used were higher in higher dose patients. So the later phase 3 trial, for phase 3 trials, I think they are using 3 milligram per kilogram dose. And there are a couple of other phase 1-2 studies on renal cell cancer. This is the one I mentioned. This is the combination of PD-1 and CTLA-4 inhibitors, where they show after 6-month progression-free survival is 65%. Overall response rate is 45%. This is quite high. And also there's another one combining nivolumab plus sunitinib or pazopanib. Unfortunately, with combination with pazopanib resulted in too many hepatic toxicities and it was stopped. But with sunitinib, overall response rate is 52% and median progression-free survival is 49 weeks, which is also long. And this is a PD-L1 inhibitor, which also shows about 20% response rate. And and in renal cell cancer, now from these data, there are 2 phase 3 trials ongoing for PD-1 inhibitors. One is use of PD-1 inhibitors versus everolimus, mTOR inhibitor, in pre-treated, so as a in the second line setting, which makes sense because everolimus is listed as one of the drugs to be used in the second line settings. And there's another one on PD-1 inhibitor combined with CTLA-4 inhibitor that shows significant activity versus sunitinib in first line setting, both of these trials are now open, and this one is already finished accrual, so the data would come in a few years' time. And as for PD-L1 inhibitors in cancer, renal cell cancer, there was another phase 1 study which showed objective response rate of 14%. So in renal cell cancer, somehow, right, presently PD-1 inhibitors is going forward rather than PD-L1 inhibitors. I will talk to you later, but in bladder cancer, it's kind of the opposite right now. Moving on to bladder cancer. So this is the study I mentioned, the phase 1 trial studying 68 heavily treated metastatic bladder cancer patients treating with PD-L1 inhibitor. It was reported in Nature last year. And one of the authors was postdoc working with Dr. Grieve, and was also working with me. But he's now an oncologist in France. So if anyone's interested in these PD-L1 studies, we have someone there who's really involved in this kind of studies. And patient characteristics. So about half of the patients were in ECOG 1 status, not ECOG 0, but had some problems. And about like 30% had liver mets, and many had visceral metastasis. And almost more than 90% had prior chemo, and many had been treated with more than 2 systemic regimens. So these patients are the patients who have no other treatment options. And this is quite striking. They did, prior to treating them, they did immunohistochemistry for the original tumor, not necessarily the metastatic tumors, but they checked the expression of PD-L1 in tumor cells as well as in tumor infiltrating lymphocytes. Indeed, I will talk a bit later, but indeed they, even though the rationale for treating going into clinical trials in cancer was that PD-L1 expression in cancer cells correlate with survival. When we look at the response of these drugs, they actually find that PD-L1 expression in tumor infiltrating lymphocytes correlate better with activity of these drugs. And about of these 70 patients, about half express IHC2/3, so high expression of PD-L1. And of those patients, 40% had objective response. This is really...
See, the 3 is still 5%. So it's really still very, very low.
Yeah.
So I don't know how valuable this data is, whether it's the actual
but I'm not sure if I don't know, but IHC2, it's a bit higher, so it's still phase 1. So I think for phase 1, this is good enough to move on to phase 2.
It's hard to gather much
Mm-hmm.
information.
Yeah.
When you talk to companies, high expression is still 5%.
Overall, but generally, I think in many cancer types, if you classify births by high versus low, so, and with...
So you're using that
No.
trial?
Yeah,
It's a
there's
post-hoc-
yeah. Mhm.
It's a randomized
Yeah.
strategy.
Mhm.
Not using it to
Yeah.
exclude.
Because the reason is that even if there's not much expression, still 10% responded. So right now, this can't be used to determine who should get the treatment or not. So overall about 50% of patients demonstrated response tumor in tumor burden as measured by the RECIST. This is if this is reproduced in phase 2 or phase 3 data, this is really quite striking. Although this actually seems a bit too good to be true data. And this is the typical response. This large lymph node disappears, and this is the waterfall plot. So these patients below this dotted line are the ones who had objective response. There are still many stable disease, which might, if in phase 3 trials, these patients might contribute to overall survival benefit as well. And also, it's again very durable. 16 of the 17 patients who responded were still continuing with treatment at data cutoff point. So again, as with other cancer types, these treatments are durable. So these data prompted the FDA to grant breakthrough status. And I will just skip on this side effect profile. It's similar to what has been reported. And now, as of January this year, a new phase 3 trial has opened. A phase 3 trial looking at this medication compared with any chemotherapy in second-line setting. So patients failing one platinum-containing chemotherapies are eligible, and estimated enrollment is about 760 patients, and accrual will end in 2 years. So after that, by 2020, the data will be many data will come. And there are some other phase 2 studies ongoing as well. Somehow for the same drug, they were opening the phase 2 trial prior to that phase 3 trial. So it seems to be running in parallel, phase 2 and 3. And there's another one using gemcitabine, cisplatin, plus CTLA-4 inhibitor as for in the first line setting. Accrual is almost done, so the data will come in a few years' time. And another one that I've mentioned, combination of PD-1 inhibitor and CTLA-4 inhibitor in bladder cancer as well. What about PD-1 inhibitor in bladder cancer? There's also phase 1 data for PD-1 inhibitor as well. Objective response rate is about 24%, and about half of them was complete response. So somehow they managed to skip the phase 2 trial, and now they have a phase 3 trial open where they you are using this medication in a similar setting, comparing against conventional chemotherapies, and this is open as well. Milk? Published? Publication, I haven't really seen this. I got this from the company as a baseline data for this phase 3 trial. So in summary, many cancers express PD-L1 and acquire immune tolerance, and targeting this axis achieved long durable response in generally about 20% of advanced, very advanced solid tumors who are otherwise untreatable. And many clinical trials are ongoing, and there are many combinations. One rationale is that it's known that many therapies, including chemotherapy or anti-angiogenic therapies, are known to induce PD-L1 expression in cancer. So they are now looking at how combination works to suppress better. So there are many combinations going. And on the topic in the research field is finding a good predictive biomarker, because we know 20% definitely have very good response, but we don't know who they are. So that's the big concern. And I would like to finish up here, but I'm sure people will ask, someone would ask, so I just have this last one slide. We know that bladder cancer, if we talk about immunotherapy in bladder cancer, people will think about superficial cancer, bladder cancer. So actually there's no real data. I couldn't really find good data here. There was just one basic research paper showing that PD-L1 high tumors had higher percentage of patients fading BCG. I'm not sure, and it's just one paper in cancer I could find, but they are actually planning a phase 1 study, which is not open yet. Oh. 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 Okay, yeah, so this is it for my talk.